Hello, we're going to present about monoclonal antibody production. We'll explain how monoclonal antibodies are made, compare the types of cells used, and look at engineering challenges and solutions from 3 scientific studies. Monoclonal antibodies are lab-made proteins that bind to a specific target, such as a virus or a cancer cell. They are used in treating cancer, autoimmune diseases, and infections. Since they are produced by living cells, not chemicals, their production requires tight control. With global demand rising, we need production systems that are scalable, safe, and cost-effective. To produce monoclonal antibodies, scientists insert the antibody gene into a host cell. The cells are cultured in a bioreactor under controlled temperature, oxygen, pH, and nutrient levels. After growth, the antibodies are harvested and purified. Choosing the right cell type is critical for large-scale success. Hybridomas were developed in 1975 by fusing a B cell with a myeloma cell. They can produce one antibody type continuously. However, they are too fragile for industrial bioreactors and less scalable. In one study, hybridoma cells were grown in stir tank bioreactors but were damaged by shear stress. Researchers replaced them with wave, hollow fiber, and cryogel bioreactors to reduce stress. This showed hybridomas need gentle, low-shear environments to survive. CHO cells are the current industry standard due to their robustness and compatibility with human proteins. In one study, media and antifoam combination caused toxicity and poor growth. Researchers used 48 mini bioreactors to test 25 conditions and identify safer options. This showed how even small components can affect the production. Plant cells like BY2 offer advantages like safety, lower contamination risks, and potentially lower cost than mammalian systems. In one case, the culture became highly viscous at scale, making mixing and filtration difficult. Researchers used a single-use orbitally shaken bioreactor and a custom purification method. This showed that plant systems work, but need specialized equipment.
Running bioreactors at scale depends on choosing the right system for the job. Hollow fiber bioreactors support long-term, high-density antibody and cell production. Cryogel reactors are useful for long-term perfusion and continuous protein production. Wave bioreactors are commonly used for flexible scale-up of CHO, hybridoma, and insect cells. Let's zoom in on the SP200X. The researcher used to test whether BY2 plant cells can produce the M12 antibody at a 100-liter scale just as efficiently as in 200 milliliter shake flask culture. The setup involved a 350-liter disposable bag filled with 100 liter of BY2 culture, shaken at 80 rpm with 5 cm orbital diameter and aerated with 20 liter per minute of air through the headspace at 26 Celsius. The process was run in the dark. The orbital shake creates a gentle, uniform mixing motion that improves oxygen transfer and nutrition distribution throughout the culture, which is essential for high-density plant cell suspensions. The system is a single-use and plastic-based, so it reduces the need for cleaning and validation, saving time and cost. The viscous BY2 broth made standard filtration ineffective, so the team used dilution and advanced techniques like expanded bed absorption and protein achromatography for downstream purification, concluding with gel filtration to purify the M12 antibody. The final fresh cell weight in the SP200X reached 387 gram per liter compared to 300 gram per liter in the shake flasks. The M12 titers were nearly identical in both systems, 18.2 mg per liter in the SP200X and 19.7 mg per liter in the shake flask. This proves scale-up did not compromise productivity. For the oxygen profile, both systems matched for the first 120 hours. After that, the SP200X dropped steadily to 0% dissolved oxygen by 149 hours due to higher cell density and oxygen demand, whereas dissolved oxygen in shake flasks dropped from 100% to 52% between 140 hours and 144 hours, aligning with the height of the exponential growth phase, and then recovered to 72%, indicating a slowdown in cellular metabolism. Still, final antibody yields were not affected. In summary, the SP200X enabled a 200-fold scale-up while maintaining growth performance and antibody productivity, making it ideal for early-stage production. However, oxygen limitations and downstream complexity must be managed. Across all articles, it's clear that each bioreactor system addresses only parts of the production chain. While single-use systems like SP200X simplify operations, they also face oxygen transfer limits at scale. Meanwhile, plant systems risk glycosylation concerns which may affect therapeutic efficacy, so success requires a comprehensive end-to-end strategy. One emerging alternative is engineered algae in photobioreactors. Algae grow quickly, can produce complex protein, even immunotoxins, and offer low cost and scalable expression. However, they lack in the glycosylation, and no large-scale GMP algae facilities exist yet. So while promising for early-stage or non-glycosylated products, algae still need development before replacing platforms like BY2.